This is week 9, genetic variation in populations continued. The learning objectives for this week are to give examples of genetic disorders occurring at high frequency in particular populations, to define the concepts of genetic drift and founder effect, to use the Hardy-Weinberg distribution to calculate the relationship between gene frequencies and genotype frequencies to determine genetic risk to individuals, and to describe ancestry testing. Natural selection is the major force driving allele frequency change. It's the chief mechanism for transforming populations, and it's the principal force that shifts allele frequencies within large populations. It was actually discovered independently by two individuals, by Charles Darwin and Alfred Russel Wallace. In looking at natural selection, we see that the concept revolves around the facts that individuals exhibit variations in phenotype and that these variations can be passed on. They're heritable. Organisms tend to reproduce in an exponential fashion. Therefore, more offspring are produced than can survive. And some phenotypes are more successful at survival and reproduce at higher rates. As a consequence of natural selection, populations and species change. The phenotypes that confer improved ability to survive and reproduce become more common, whereas the phenotypes that confer poor prospects for survival and reproduction may disappear. Populations may no longer be able to interbreed. The result may be the appearance of a new species. Allele frequencies may change from generation to generation if all the genotypes do not have equal rates of survival and they do not leave equal numbers of offspring. This shifts the allele frequencies within large populations, and it's one of the most important agents of evolutionary change. In looking at Hardy-Weinberg analysis, it allows fitness to be examined for each genotype. Fitness is the individual's genetic contribution to future generations. Homozygous recessive individuals who die before producing offspring have an F equal to 0. The frequency of the recessive allele will decrease in each generation. This slide is showing the change in the frequency of a lethal recessive allele, lowercase a. The frequency of the gene is halved in 2 generations and halved again by the 6th generation. Subsequent reductions occur slowly because the majority of the alleles are carried by heterozygotes. This next slide will show the outcome of different degrees of selection against a non-lethal recessive allele. In this case, the intensity of selection varies from strong, which is the red curve, to weak, which is the blue curve, as well as in intermediate values of yellow, purple, and green curves. In each example, the frequency of the deleterious allele starts at 0.99 and declines over time. However, the rate of decline depends heavily on the strength of the selection. And as you can see in this slide, the effect of selection and what it has over allele frequency, the rate at which the deleterious allele is removed from a population depends heavily on the strength of selection. So look at this, please. If you look at selection, it can be classified as directional selection, stabilizing selection, or disrupted selection. If we look at directional selection, this is where the phenotypes at one end of the spectrum become selected for or against. And what happens is this usually is a result of changes in the environment. Let's give an example. If we go back to Darwin, is that the beak sizes in the finches during the dry years increased due to strong selection. This slide illustrates the beak size in finches during dry years increases because of strong selection. Between droughts, selection for large beak size is not as strong, and birds with smaller beak sizes survive and reproduce, increasing the number of birds with smaller beaks. In looking at stabilizing selection, intermediate types are favored. Both extreme phenotypes are selected against. It reduces the population's variance over time, but not the mean. An example of this is a human birth weight study over an 11-year period. In looking at this slide, it shows a distribution of birth weight, the percentage of mortality at 5 weeks, and the percent of births in the population at right. Infant mortality increases on either side of the optimum birth weight of 7.5 pounds. Stabilizing selection acts to keep a population well adapted to its current environment. In disruptive selection, both phenotype extremes are selected for, intermediates are selected against. This results in a population with increasingly bimodal distribution for trait. I can give an example with Drosophila for applied selection for low and high bristle number. In this slide, the effect of disruptive selection on bristle number in Drosophila, when individuals with the highest and lowest bristle number were selected, the population showed a overlapping divergence in only 12 generations. Mutation creates new alleles in a gene pool, and within a population, the gene pool is reshuffled each generation. It's the only process that creates new alleles in the gene pool. Most mutations are recessive. Indirect methods using probability and statistics or large-scale screening programs are often employed to estimate mutation rates. Changing allele frequencies, several conditions for accuracy must be met for accuracy when measuring significant force of changing allele frequencies. The allele must produce distinctive phenotype that can be distinguished from similar phenotypes produced by recessive alleles, and the trait must also be fully expressed or completely penetrant so that mutants can be identified. Identical phenotype must never be produced by non-genetic agents or chemicals. In humans, a dominant form of dwarfism known as achondroplasia, which we've discussed before, fulfills the requirements for measuring mutation rates. Individuals with this skeletal disorder have an enlarged skull, large arms and legs, and can be diagnosed by X-ray examination at birth. In a survey of almost 250,000 births, the mutation rate for achondroplasia has been calculated. And looking at mutation rates, it's the number of new mutant alleles per given number of gametes. And if the mutation rate is known, the extent of change to allele frequency from one generation to the next can be estimated. It is important to note that non-random mating can change frequencies of genotypes in a population, and the subsequent selection for or against certain genotypes can affect overall allele frequencies. But non-random mating itself does not directly change allele frequencies. Migration occurs when individuals move between populations. Species divides into populations that are separated geographically. Allele frequencies in new populations may differ over time. In looking at this slide, it's a slide on migration, and we can see that this is demonstrating migration as a force in evolution. The B allele in the ABO locus is present in a gradient from east to west. This allele shows the highest frequency in Central Asia and the lowest in northeast Spain. The gradient parallels the waves of the Mongol migration into Europe following the fall of the Roman Empire and is a genetic relic of human history. Genetic drift is the significant random fluctuations in allele frequencies in small populations. It's possible by chance alone. The degree of fluctuation increases as the population size decreases. Can also occur as a result of the founder effect and a genetic bottleneck. Genetic drift can also arise through what is called the founder effect. This occurs when a population originates from a small number of individuals. The gene pool may not reflect the larger population from which the founders are actually drawn. Genetic drift can also occur as a result of genetic bottleneck. This develops when a large population undergoes drastic but temporary reduction in numbers. Populations may recover, but with greatly reduced genetic diversity. And giving an example of a founder effect in human populations, we're going to look at the high frequency of oculocutaneous albinism, OCA, in the Navajo Indians, Native Americans. In the Navajo, who live primarily in northeast Arizona, albinism occurs with a frequency of 1 in 1,500 to 1 in 2,000, compared with a Caucasian population of 1 in 36,000 and the African Americans of 1 in 10,000. There are 4 different forms of the ocular albinism, all with varying degrees of melanin deficiency in the skin, eyes, and hair. OCA2 is caused by a mutation in the P gene, which encodes a plasma membrane protein. To investigate the genetic basis of albinism in the Navajo, researchers screened for mutations in the P gene. In their study, all Navajo with albinism were homozygous for a 1.22.5 kb deletion in the P gene, which spanned exons 10 to 20. This deletion that caused the OCA2 albinism was found only in the Navajo population and not in members of other Native American tribes in the southwestern United States, suggesting that the mutant allele is specific to the Navajo and may have arisen in a single individual who was one of a small number of founders of the Navajo population. Workers originally estimated the age of the mutation to be between 400 and 11,000 years, but tribal history and Navajo oral tradition indicated that the Navajo and Apache became separate populations between 600 and 1,000 years ago. Because the deletion is not found in the Apaches, it probably arose in the Navajo population after the tribes split. On this basis, the deletion is estimated to be 400 to 1,000 years old and probably arose as a founder mutation. I'd like you to look at some other examples of founder mutations, and this week one of your first question will be in the problem set to give an example of 2 other founder mutations that exist. Non-random mating can change frequencies of genotypes in the population. Subsequent selection for or against certain genotypes can affect overall allele frequencies, but non-random mating itself does not directly change allele frequencies. There are 3 forms of non-random mating. Positive assortative mating, which similar genotypes are more likely to mate than dissimilar ones. Negative assortative mating, dissimilar genotypes are more likely to mate than similar ones. And inbreeding, mating individuals closely related. Consanguinity is the term that's used for inbreeding. For a given allele, inbreeding increases the proportion of homozygotes in the population, and completely inbred population theoretically would consist only of homozygotes. In looking at selection in recessive diseases, as long as mating is random, genotypes in autosomal recessive diseases can be considered to be in Hardy-Weinberg equilibrium. This is despite selection against homozygotes for the recessive allele. Remember that selection against harmful recessive mutations has far less of an effect on the population frequency of the mutant allele than does selection against the dominant mutations. Because remember, only a very small proportion of the genes are actually present in homozygotes. In looking at selection and dominant disorders, dominant mutant alleles are exposed directly to selection. The effects of selection and mutation are more obvious and can be more readily measured for dominant traits. If a gene is lethal and dominant and fully penetrant, it will be exposed to selection in the heterozygotes. It removes all alleles responsible for the disorder in a single generation. Several human diseases are thought or known to be autosomal dominant traits with zero or near zero fitness and always result from new mutations. This is very important for genetic counselors. In some, the genes and specific mutant alleles are known, and family studies show new mutations in the affected individuals that were not inherited from the parents. But in other times, the genes are not known, but a paternal age effect has been seen, suggesting that a de novo mutation in the paternal germline is a possible cause of the disorder. Please remember about this when we discuss dominant disorders, with de novo mutations being associated with advanced paternal age. The implication for genetic counseling is that the parents of a child with an autosomal dominant but genetically lethal condition will typically have a very low risk for recurrence in subsequent pregnancies because the condition would generally require another independent mutation to recur. Let's look at some examples of diseases occurring as sporadic conditions due to new mutations with zero fitness. Adelotosteogenesis is one condition of which a gene has not been found yet. It is an early lethal form of short-limb skeletal dysplasia. Cornelia de Lange syndrome we'll discuss in a little bit more detail in a moment, but the majority of the cases are caused by the NIPBL gene. Osteogenesis imperfecta type 2 is a perinatal lethal type of osteogenesis imperfecta with a defect in the type 1 collagen. This can be caused by COL1A1 or COL1A2 genes. And then thanatophoric dysplasia is an early lethal form of skeletal dysplasia due to de novo mutations in the FGFR3 gene. Let's look at Cornelia de Lange syndrome with the OMIM number of 122470. It's a very rare genetic disorder. It's present from birth, but it is not always diagnosed at birth. It causes a range of physical, cognitive, and medical challenges and affects both sexes equally. It is named after the Dutch pediatrician Cornelia Catharina de Lange, who described the disease back around the 1930s. It is often termed Bushy syndrome and is also known as Amsterdam dwarfism. It can lead to severe developmental anomalies, both physically and intellectually, affecting the development of a child. The incidence is estimated at 1 in 10,000 to 30,000. The gene that is primarily responsible for about 50% of the cases is the NIPBL gene. This gene was found to be on chromosome 5 in 2004 simultaneously by two research groups, one at Newcastle in the United Kingdom and the other at Children's Hospital of Philadelphia in the United States. There are two other genes which have been shown to cause a similar disorder, SMCI1 and SMC3, but they cause a much milder form of the disease. If we look at the signs and symptoms in Cornelia de Lange syndrome, we see that low birth weight, usually around 5 pounds or 2.5 kilograms, developmental delay, small stature, micromelia, smallness of one or more limbs, and missing limbs or portions of limbs can also occur. Microcephalia and intellectual disabilities occur. Synopsis, which is thick eyebrows, which typically meet at a midline, long eyelashes, short upturned nose and thin downturned lips, long philtrum, gastroesophageal reflex, congenital heart defects, many of the different types can occur, as well as vision disorders. We also see that the children often have a lot of hair. Those conditions those symptoms which are in red are ones that you most often do see. I will hold this slide for a moment because I want you to see that these two children here do look similar to each other, and they both have Cornelia de Lange syndrome. And you can see some of the things that I was talking to you about with the long philtrum, the bushy eyelashes. You can see the bushy eyebrows, the hair, the excess hair on the body. In the case of the little boy, he has some on his forehead here. In looking at mutation and selection balance in dominant disease, if the gene is deleterious and not lethal, then the affected individuals will be able to reproduce, but their fitness, F, is reduced. Remember that fitness is the chief factor that determines whether the mutation is eliminated immediately, becomes stable in the population, or even becomes over time the predominant allele at the locus concerned. The frequency of an allele in a population at any given time represents a balance between the rate at which mutant alleles appear through mutation and the effects of selection. The deleterious dominant gene will be lost through selection at a rate proportional to the reduced fitness of the heterozygotes. The frequency of the mutant alleles responsible for the disease in a population therefore represents a balance between loss of mutant alleles through the effects of selection and gain of mutant alleles through recurrent mutation. De novo mutation. Stable allele frequency will be reached at whatever level balances the two opposing forces, selection and de novo mutation. The mutation rate per generation must be sufficient to account for that fraction of all the mutant alleles that are lost by selection from each generation. If the fitness of affected person suddenly improves, the observed incidence of the disease in a population would be predicted to increase and reach a new equilibrium. This is something that we are seeing more of due to the fact of modern medicine treating various diseases which we were not able to do before. Your textbook, Thompson and Thompson, discusses retinoblastoma in this manner. Mutation and selection balance in X-linked recessive mutations occurs because selection occurs in hemizygous males and not in heterozygous females, except for those females who are manifesting heterozygotes with reduced fitness. This completes the content for this shortened week, and you will then now have some problems to work on, as well as answering a question about two examples of genes that are part of the founder effect, that demonstrate the founder effect.